This is CheloniaCast, episode 19. Thanks for joining us today. Thanks to the Turtle Room for sponsoring this episode and show. You can find us at theturtleroom.org/CheloniaCast, where you can learn more about our program, as well as access the CheloniaCast Student Research Fund, where we'll be raising money to support student-led turtle conservation and research-oriented projects, as well as other programs dealing with turtle and tortoise biology. Today we are joined by Scott Thomson, who is currently in the Museum of Zoology at the University of Sao Paulo. He's also working at the Centro de Estudios de Colonias de Amazon in Manaus, Brazil. He got his master's in zoology and mathematics at the University of Canberra, studying with Arthur Georges. He's done a variety of he's published over 60 peer-reviewed publications ranging in focusing on systematics and taxonomy and kind of evolution of snake-neck turtles and Australian sort of Chelids. And we're going to be talking to him today about a lot of these things. Really happy to have you on today, Scott.
It's kind of a question we ask every time, but what first got you interested in turtles?
Okay. My passion for turtles actually started young. My mother was very insistent upon us reading a lot of books. And when I was extremely young, only about 6, she bought me a book called The Enchanted Isles, which was about the Galapagos. And yeah, it's a children's book, but it showed that there were different on each island. And it basically, although not in those words, it introduced me to the idea of island biogeography. And so later in life, I realized that's what it was actually showing. And it stemmed all the way through from there. I just slowly, by 10 years old, I had snake-neck turtles in captivity. I had a group of 5 of them. They were my first turtles, about in 1975. And I kept turtles up until the time, I joined the army for a few years, but after I got out of that, I got back into working in zoology again. I started with fish again after that, but then started working with Galapagos tortoises at Western Plains Zoo in Australia as a zookeeper. And they had a group of 10 tortoises, and I developed methods of identifying Galapagos tortoises to subspecies morphometrics. This was before DNA was available. And I was relatively successful at being able to do it. Many of my IDs ended up being correct. DNA was used to confirm them. But it's not an ideal method for doing it, I know. And so I was always interested in how things split up and evolved. And I got the opportunity to do that in Australia, where I lived at the time, and started working on long necks, but also I was given the opportunity to take a really good look at the short necks because they were a mess. They were, 20 years ago, their taxonomy was disaster, and I did my master's on Elsaya. And at the same time, I was also doing some descriptions of long necks and looking at the fossil history as well, because I feel the fossil history is so important in turtles, because there's 4 times more fossils, taxa of turtles, than there are living species. There's over 1,200 fossil taxa described. And so it's important to always include the fossils in a group that's got such a fossil history.
Right, that's an interesting point. That's an interesting point. The fossils, in terms of using that in phylogenetics and trying to answer questions about relationships, it's something that's not always there, and seems like it would benefit a lot of analyses if it was. Talking about the Elsaya, maybe we could backtrack to that. That seems like we could start off with that. That was sort of an interesting study there, I think, beginning with some of the Alzheimer's work that Dr. Georges did to kind of discover all these different haplotypes or groupings, and then you came through and over 20 years or so described a bunch of the new species there. Maybe you could shed some light on the process behind that.
Yeah, it technically started back in 1974 with Andrew Burbidge, who did serology, which was what was available back then. And he found some divisions within Elsea and also the longnecks that needed further testing. And Arthur Georges with Mark Adams, he did the Allozyme work, as you said, and he went on to show that there were at least 5 taxa within species dentata, and that what became Myochelys was not really an Elsea. You've got to remember, back then we had 3 species of Elsea and that were recognized: Elsea latisternum, Elsea dentata, and Elsea miniae. That was it. And he brought me on as a master student to actually do the morphology, the morphometrics, and the descriptions of the various species. And we went from there. I ended up describing with Arthur, or at least the 3 of them, 4 species of Elsea, and I also split the genus Elsea. And so, and as I said, we had 3 species. By the time I finished, we had 11 species in one genus and 4 species in another. John Can described Irwini, so I didn't describe all of them, but I did describe 4 of them.
Yeah, and which 4 were those again?
I described Elsea albigula with Arthur Georges and Colin Limpus. I also described Elsea rhodoni with and Yolani, who's from Papua New Guinea. And I described Elsea flaviventralis. And I also described a species called Elsea nadabajagu. And the other thing we did was deal with some species that had already technically been described but had been sunk. So I resurrected Brandohorsti, I resurrected Schultzi, I also resurrected Uberima, corrected the usage of Telemastes, and that's why we have the 3 subgenera within Elsea that possibly should be genera, honestly. So Elsea is up for more splitting if you want, because Pelecates and the true Elsea are actually nothing alike. They're completely different animals. And this would go for Hanmerichelys as well, which is the New Guinea stream turtles.
Right. That's pretty interesting. And so you kind of came through after the Allozyme work was done and then characterized these haplotypes, but using morphology. Maybe you could shed some light on what morphologically, what are the things we're looking for that kind of denote differences between these turtles?
All right. One of the problems in morphology that a lot of people do is they only look at live animals or something like that, and they only look externally. And that's a difficulty. You need to actually get skeletons and look at how they differ skeletally, because that tells you a lot about their function or morphology. And function is important. The Pelecamastes, they differ in the form of the alveolar ridges within the mouth. They are true fig eaters. They don't eat anything else really. And so their alveolar ridges are designed to shred fruit. This is a whole different formation of the palate within the turtle. And they also differ in the form of what I ended up with a bridge strut. A lot of people call them buttresses. I don't do that. Among my degrees, I have a degree in computer science. I'm a software engineer, and that meant I did engineering. Engineer, I look at structure from an engineering perspective. And what people are calling a buttress is not actually a buttress. From an engineering perspective, it's a strut. And because buttress only prevents movement in one direction, one plane direction, whereas a strut can prevent movement in three directions because of its angle. And so what people call the buttresses in turtles are actually struts from an engineering perspective. So I renamed all of that nomenclature within. I'll show you what a bridge strut looks like, if you like. This is part of the plastron. So if you look in there, this is the bridge strut. All right, this part here. There's the bottom. Okay, that's the front of the turtle. That's the side. And as you go in, that's the inside. And it basically, that is the bridge strut.
And so that's a diagnostic feature. You
And
look
that
at the angle of
basically
that
is what connects the plastron, and it is a in river turtles it's very big because it's used to stop crushing of the face. Basically, it's to stop crocodile attacks. So wherever you've got big living with fresh water or saltwater crop, something like that, or any sort of animal that can crush a turtle, you'll end up with turtles with big struts. And it's not just in Chelid, it's found in Bataga and some ammidids and things as well. But whenever you've got a large river turtle, you'll end up with these big struts that create extra strength within the shell to prevent crushing.
So you use kind of the bridge struts, the angle of the bridge struts and the shape, right? Is there anything else like anatomically that you're using to classify the Alcea, or is that?
Yeah, there is. There's quite a lot. I mean, I actually have 63 characters, morphological characters for Alcea and Macrocephaly and Embryonic, so for the short necks. And there's things like the first vertebral scoot in Alcea is very consistently wider than the second, and whereas in Embryonic and Re-Substitution they're the same. Same with Macrocephaly, they're the same. And we also have characters like even the posterior bridge strut, where it actually comes into the carapace is different. It is either at the fifth and sixth, literally between the fifth and sixth plural, or it can be on the fifth plural, or it can be on the sixth plural, depending on the genus. And then there's characters like the position of the vomer, which is one of the bones on the base of the skull. Hang on 1 sec, I'll get one.
One of the palatal bones, right? The palatal complex.
All right, this is a Frenox, but if you look,
Oh, there it is.
Oh,
yeah, that bone between the two holes, the one that's below it, that's the vomer. And either side of the vomer are the palatines, and you get it, pen. And then further down are the pterygoids. So, right, sorry, I've got a pen here now. Point. That's the vomer. And how that bone is connected to the palatines or the pterygoids is different between species. And that's to do with how they eat, actually, and how much strengthening they need inside the jaw for eating. And so it depends on what they're eating, etc. Also use presence, absence of a nuchal scoot, believe it or not, works for distinguishing Hanweracheles from all the others, because all Hanweracheles have nuchal scoots. True Elsea and Pelecomastes do not. The very front of the carapace in Pelecomastes is not quite, but almost indented, a bit like podocnomids are, whereas in Elseas they're very rounded. And colour in Flaviventralis has a yellow belly. It's the only Elsea with a yellow belly. All the rest of them have black, or at least mottling to black on the belly. Yeah, so there's dozens and dozens of characters.
Right. There's a lot. That's probably a good thing when you're trying to create a robust or I guess anatomically based phylogeny or classifications, if you can distinguish a lot of things.
Yes, you want a lot of characters. I mean, I may end up only using 3 or 4 characters when I write the key for how to distinguish them between species for people with live animals. But for the actual phylogenetic analysis, I want at least 50 to 100 characters. I use actually around 230 characters now.
Wow, that's a cool thing. And that's pretty interesting. And the question here a lot of times, right, is distinguishing between species in allopatry. For a lot of listeners, that biological species concept of if they can kind of produce fertile offspring or not with each other is going to be the idea of what we have of species. But there's a lot of different species concepts, and it's not always that straightforward if you can tell if something can produce viable offspring, right? So you're using these characters under what sort of context?
Well, we're using the characters in terms that they show diverging evolutionary trajectories. So obviously the characters have functions, and the turtles have developed different ecological niches that they're using, so they have to have different morphology, so they diverge, and that's how you end up with different species. Hybridization is an issue. Back in 1963, when the biological species concept was proposed by Mayer and Simpson and co, the ability to track hybridization wasn't possible, really. I mean, nowadays we can use molecular data to identify hybridization, or what we would generally call introgression, where genes are being flipped between species. And what it's shown is that hybridization is virtually normal. And let's be honest, even our own species is a hybrid. There are 3 taxa inside our species, and different humans from different parts of the world have between 2 and 6% of other species inside of us. So, hybridization is normal. And with turtles, they're very good at it. I mean, every Chelidina can breed with any Chelidina. I've got photographs of a Chelidina parkeri cross Emodura subglobosa. And so that's cross genus, short neck over long neck, bred with each other, no problem. And what this does for species is it actually allows them to import genes into their genome that they can possibly use to adapt to changing conditions. And so the species is then using those in the adaptation, and so you end up with a history of introgression. And so hybridization is not rare, and it's basically pretty normal. And we need to start using that or at least accepting that within our species concepts, that hybridization occurs, probably across most taxa.
Right. The idea, the evolutionarily significant unit kind of encapsulates that idea, right? That things are, a species should be something on an evolutionary trajectory, independent of whether it can interbreed with other things. That definition is kind of shaky as well, right?
Well, there's around about 22, 23 different definitions of a species floating around, and they're all pretty shaky. Not so much because you can find a group of taxa where a good species concept will work for that group of taxa. But if you want to have a species concept that'll work for everything from a fungus to a vertebrate, you're going to be in a lot of difficulty because you've got different reproductive modes there. You've got asexual and reproduction, all sorts of things. Plants can reproduce often in 3 or 4 different ways, by budding, by fruit, within the same species. So there's all sorts of issues going on that make defining a species exactly very difficult. So what you really want to look at is, to me, you want to look at whether or not, as you said, is it on an evolutionary trajectory that has diverged enough from other trajectories that they are different enough to define them at both a morphological and a molecular level.
Right. That makes sense. I think too, something interesting for all of us as kind of younger, kind of getting into this field, it's cool to read papers that describe new species, because there typically is a lot that you won't find in terms of describing it in a holistic manner, but also the details of the ecology for a new animal, a new taxa. What is it like? Like, what is the process and what kind of analytical methods are you using to validate some of these things, right? Because you can go into a population and measure different characters, but that doesn't necessarily mean that's going to hold over a large range. Or how do we statistically kind of buffer the assumptions of speciation?
All right, so the process of describing a species is complicated, and what you basically need is to look at the weight of evidence. And I mean, like you say, I can look at some population and say, that's a new species. Okay, prove it. And that's what you've got to do. You've got to get this weight of evidence that effectively proves your hypothesis. So you've got a hypothesis with a null hypothesis. This is a new species. No, it's not. And you need to get this balance of evidence to get you to the point of saying it is. And this is a problem with some of the species descriptions that come out. They don't produce any real evidence. They just say it's a species, but the evidence is not there. So what you want is multiple lines of evidence, for one, that corroborate with each other. And so I will not describe a species based on a singular line of evidence. So if I've only got morphology, that's not enough. If I've only got molecular, not enough. If I've only got morphometrics, no way. I use all three. Obviously with fossils, I can't. They're rocks, let's face it, they don't have DNA. And, but I still use morphometrics and morphology and also a few other features in fossils which I can't use in living taxa, but such as taphonomy and age. Age does come into it with fossils, but with the living taxa, I will say, for example, I'll say Albigula. We had some evidence that it was a new species before I started. Arthur's Allozyme work, for example. It had also been suggested to be a new species by John Cann at one point. Before him, even back to the 60s, people had 1960s, people had said this might be different. But the allozymes by themselves were not enough, even though it was a significant difference in allozymes from dentata. I then had to produce the morphological evidence that they were different, all those characters that we were discussing before, and then a whole bunch of measurements. And when I'm doing measurements, I'm not just measuring shell length and then shell width and putting one over the other and saying this is different by a simple statistical method. I'm using principal components analysis followed up by a discriminant function analysis. And what that does, principal components analysis, will basically throw all your data in and it'll spray it out all over the graph, and hopefully it clumps into groups. And you can see that there's, say, 3 different groups that are very, very different. And that might be dentata, albigula, and irwinite, for example. I then take those groups and I classify them. And I say one is species A, one is species B, and one is species D, whatever. And put them back into a new analysis called a discriminant function analysis, which uses classification and ordination. And that reanalyzes it with constrained classes to show the difference between those classes. So now I've got a morphometric analysis that supports the hypothesis. I have a morphological analysis that supports the hypothesis, and I have allozymes, which is as good as molecular in a lot of ways at the time. And they all supported each other. And so I have 3 independent lines of evidence that support the hypothesis. Then I can say it's a species. And that's the process, basically.
It's a pretty interesting confluence of a lot of different, like mathematics and biology. I mean, it's pretty interesting how you can take all of these different characters and then use these analyses to look at something that exists in a high dimensional space and then kind of boil it down to something that you can even cross validate, right? You can take taxa measurements and then it will sort them into different categories, and it gives you a percentage score for how similar they match those canonical space, right?
Yeah, this is why the discriminant function analysis is so important as well, because one of the features of the discriminant function analysis is that it actually attempts to destroy itself. It does what's called stepwise analysis. So every step, it is removing one data point and seeing if the analysis still holds. And it does another step, sees if the analysis still holds, removes another variable, sees if the analysis still holds. And you see how far it can go before it fails, as in you lose your 95% confidence limit. It will then show you that. And the further it can get without being self-destroying, the higher the p-value is of the analysis, the probability. And the next thing it does is a re-substitution analysis. It reanalyzes the entire thing again with one specimen missing out of the data. It then tries to classify that specimen just using the mass without the class added and sees if it gets it right. And you have what are called priors, which is your minimum amount of specimens that must be classified correctly for you to accept the analysis. And if it keeps getting all of them correct when it's trying to predict what species it is, then that adds to the value as well. And this is why I use that type of analysis. It's a very, very powerful analysis. And just doing an ANOVA or something like that, or a simple chi-square or anything, is just not enough. It's very basic statistics for this type of stuff.
Right. It's not as dynamic as that system. And it seems like, I recall the description of the Pearl River map turtle. They used that, but it was the first time I'd actually seen it used in North American turtle classifications. So it seems like a good thing to do beyond just your simple kind of, I guess, multivariate comparisons.
Yeah, it is. You need to take your statistics as far as you can go. You need to meet all of your assumptions, because every statistical test has a series of assumptions that you have to meet to use the test. So you have to meet those assumptions. And so you've got to do the stats right. Same with the phylogenetic analysis. They also have statistics. involved in the tree, which is your consistency index, your retention index, which are statistical tests of the analysis. So
Right.
you also have p-values on that.
That's interesting. That might be a good time to sort of transition to just Chelid taxonomy in general, and even kind of biogeography. I think one of the things that stands out about this group of turtles is the fact that you've got them in Australia and South America, and then nothing in sort of the northern hemispheres. It's a pretty interesting biogeography. Maybe you could sort of give us a little bit background on why this is, like what are the sort of the events that have led to this, and then on their phylogeny, if there's any one phylogeny that's currently accepted, how has this happened through the genera?
Yeah, Chelids are a extremely interesting group because they're entirely Gondwanan. So they're only found in the continents of Gondwana, and in particular South America and Australia, as you said. They're also almost definitely found in Antarctica, were fossils. And what happened is that up until 103 million years ago, Australia, Antarctica, and South America were connected. And the chelids were likely distributed right throughout that whole area, but the southern part of South America, southern part of Australia, and Antarctica. Once those three continents split up and Antarctica froze, obviously they went extinct in South America, in Antarctica. And they spread a little bit further north in South America and Australia. They're actually a cold climate turtle, very interestingly. Chelids are still active in water that is only 12 degrees Celsius. I don't remember what that is in Fahrenheit, but it's very cold. I've been in water that cold, catching turtles, and you go very blue, I can assure you. It's not fun. They're completely active in that water and still hunting for food. They're actually not very good in tropics, and there are not a lot of tropical expansions of the chelids until very recently. And what we have is a group that basically, based on the fossils, evolved originally in Argentina. And they seem to have evolved around 145, 150 million years ago in Argentina, and then spread south down through Antarctica and across to Australia. They probably evolved. There's a number of phylogenetic analyses that actually support the idea that they are the sister group of a Palaemodozoides group called the Herpetologist. And they are an extinct group of long-necked turtles from the Palaemodozoides. And if that's correct, what chelids are is actually just another Palaemodozoides. And they're just a very different one. And personally, I think that is actually highly likely. And because the Herpetologist are also from Brazil, and including further south all the way to Argentina. So they are actually in the right place, they're at the right time, and morphologically they have a lot of similarities. So yeah, to me the chelids evolved in the south and they stayed there. They never got across. You've got to remember all of these southern Gondwanan continents were not connected in any way to the northern continent until very recently, when South America collided with North America, for example, when Australia got close enough to Indonesia to start having some crossover there. But historically, they were only found in the far south.
Do you think that the fact that they aren't as they don't prefer the tropical conditions as much, like limited their ability to move northwards on the Great American Interchange, like, you know, or like some species came south, like that's why there's Chelidra in the northern parts of South America, but there are no chelids or anything that moved up north into North America?
I would say partly that, and also probably also a bit of competition. They probably didn't compete well with the amidids coming down. Various trichemi species that moved down through that into northern South America probably didn't help them as well. And but yeah, they never got any further north than probably the furthest north they are is Bermuda. That's it. But honestly, I think they're possibly human introduced anyway. There are matters in parts of the Caribbean. I think they may be human introduced.
Where did you say specifically? Which?
There's a couple of islands in the Caribbean where you can find matter.
Oh yeah.
But Barbados,
Doesn't make a
I
lot of
think.
sense. Like,
I think they're human introduced. I don't think
Yeah,
they're
but-
naturally. They've been there a long time. Don't get me wrong. I don't think that's a recent introduction, but they
I
probably
think it's
are.
like prehistoric humans.
Well, certainly several 100,000 years, yeah, or 30,000 years, or something like that.
Yeah.
I was reading over the paper by Hawley that used the like total evidence matrix for constructing the dated phylogeny for all the Chelids. It seemed like they had the Araripemidae as basal to the other Pelamedusoides, and then that was like a whole separate. So that would be against the hypothesis. So you're saying that Chelids could actually be a radiation of the Araripemidae, which is kind of it's a basal group to the Pelamedusidae, essentially. Is that?
Yeah, it's a very ancient group, and it is certainly basal to a number of taxa. So it's a stem. It's
Right,
a
stem. Yeah.
But I'm not 100% in agreement with Holly's phylogeny. I don't think he got all of it right. He did try very well. He did a good job with a lot of it, and it was very interesting. But there are other phylogenies that have been produced that even have Araripemidae as sister taxa. But I need to go right into their data to see how they got that. Because one of the problems in Chelid phylogeny is people misinterpret long neck. They use long neck as a character. And that's a misinterpretation of how to use that character. And if you're just using long neck, it's going to create conjunction where there's homoplasy. And homoplasy being, the same character evolves in two different organisms independently. That's homoplasy. So the interesting thing is, I think the thing that's homoplasic in chelids is short neck, not long neck. I think the short neck has evolved multiple times, not the long neck.
So the long neck would be ancestral. That would be what the ancestral Chelid had, and then you had the short neck radiation in both South and Australasia, right? South American,
Yeah,
Australasia.
That's what I think. And I think one of the big hassles in Chelid phylogeny is the root of the tree. They're rooting it on Podoctomus, which is a short neck. And then they're using long neck as a character, which makes all the long necks come together in a morphological tree. But if you root the tree on Araripemes, you'll get the opposite. Because that's how the algorithm works that you use in
Right,
phylogeny analysis. So you've got to be extremely careful what you root the tree on to get the phylogenetics to work. And it takes a lot of understanding of the anatomy.
Right, that makes sense. Like you said, the morphology, the long neck state, if it's homoplasias or not, depend. Like the genetic analyses seem to support the idea that it would be right, whereas morphology doesn't, because it links them all together. That's pretty interesting. What do you think that the functional significance of? Well, do you think it's homoplasias or not?
Well, it's homoplasic, but of course it depends on where you look at the tree. At some point, it was also a derived state, because Araripemes has a long neck, but no other Pelamedusoides does. So obviously, it evolved a long neck for a reason.
Right.
And if you go that far back in the tree, it is a derived state. But from then on, it's an ancestral state for the species that evolve after that.
What do you think that the functional significance of the short neck evolving in two different continents at two separate times would be? Is that
food sources? Basically, you're looking at the short neck has a lot of advantages if you're a sedentary feeder. So basically, if you're herbivore, if you're eating mussels or snails or anything like that, you don't need a long neck. And the long neck has a disadvantage for predation, especially, for example, eagles catch long neck turtles by just grabbing them by the neck because their necks hang out. There's not as much protection at the front of the shell. So there is a disadvantage to the long neck, and the long necks seem to have been a successful concept amongst the evolution of these turtles, but it has its limits, and so when opportunity arises, the neck goes back to short neck. But some of the what you would call a short actually have extraordinarily long necks. I mean, it's not short neck, long neck. It's really not that simple. And personally, I don't consider the matter, for example, a long neck. I consider it a short neck. There are reasons for that, which are very complicated. But I'll show you one here. This is Frenox, Chefroana. Okay.
There we go. So, oh yeah, it's alive. Okay, it's alive.
Yes. Can you see its neck?
Yes.
That is not just a short neck. It has a relatively long neck.
Yeah, right. That seems like.
No Elsea, no Myakelles has a neck that long. Not even.
Oh, here comes another one.
That's
wow, rhinomers.
Whoa, yeah, I've never seen one of them.
So this guy has a short neck. That's a short neck. But Frenox's neck is twice the length of that. So is Frenox a short neck or a long neck?
Well, it's still I think Hydromedusa are the only ones that are considered long necks, right, in South America, but that doesn't seem right.
Well, Chelid has a relatively long neck, but it's not as long as Hydromedusa or the Australian Chelidinus. And the thing with Chelid is there's a bit of an optical illusion going on when you look at Chelid. Chelid is an extremely big turtle, but it has a very small shell. Yes, a 60 centimeter Chelid has a big shell, but a 60 centimeter Chelid would be the equivalent of an 80 centimeter Frenox because they have less overhang of the shell compared to the length of the vertebrae. Their shell has shrunk. Their bridge length is actually shorter, and the bridge only goes from the junction of the around about the third marginal down to about the sixth margin, whereas in most other chelids goes to the eighth. And so the whole bridge has shrunk in Chelid. All the overhang of the carapace at the front and the back has reduced. Its tail and legs hang out like you wouldn't believe compared to other chelids. Its plastron is so short that the pelvis is connected to the very butt end of the plastron. And that's one of the other factors with that group. So Chelid, I always describe it to people as having a short shell, not a long neck.
I think that's a good point, right? It's more of a gradation in some ways. There's not, when you really think about it, Phrynops, the chelonians, they're gonna. It's nothing in North America has a neck that long. It's not really a short neck. It's kind of just this gradation.
Yeah, and also with Chelid, because the shell's so short, more of the neck is exposed. The last joint of the cervicals, which is your actual neck bones, is closer to the front of the shell in a Chelid than it is in any other chelid. So more of the neck is exposed, making it look longer. So it's an optical illusion.
Interesting. So when you actually take the measurement and then control for size, it's similar. Yeah,
what you need to do is, instead of measuring shell length, you measure the length of the thoracic vertebrae and compare that. That's your baseline. The thoracic vertebrae becomes your baseline. You get shell length as a variable. And when you do that, you find out that the matter actually has a very short shell relative to vertebrae length.
That's pretty interesting. So a lot of people were wondering too about what causes the long neck condition. What are sort of anatomically, is it just elongation of the cervical vertebrae, or are there other things that are happening there that lead to that?
There's quite a lot. Obviously, the cervical vertebrae are elongated, and leave callus out of this for a minute, because they actually break all the rules on this. When you actually break it down into individual bones, they are not like this. But in Chelidina and Hydromedusa, you've got complete fusion of the atlas axis structure and elongation of it. So it actually creates like an extra vertebrae in the neck. Rather than it just being for connecting the head to the neck, it also is elongated and fused. So that creates this extra vertebrae. And then all the other vertebrae are elongated except the eighth cervical, which connects directly to the thoracic vertebrae inside the shell. And that's where you get the length from. And in Chelidina expansa, for example, from Australia, the fourth cervical is twice the length of the eighth cervical. And that creates a huge amount of length in the neck. So fully grown Chelidina expansa, although 40 centimeters in parapet length, is a 1 meter turtle from nose to tail.
It's interesting that's the case. So the ribs are also kind of structurally interesting in certain species to make more room for the longissimus dorsi muscles. Is that a condition that's different among chelids, or is that something that's fixed?
The ribs, which, sorry?
The ribs are kind of modified in a way that in certain species makes more room for the longissimus muscles, like differential development.
Yeah. All right. Let me 1 second, I'll get a specimen.
Okay, sounds good. I like this. We're getting the full on specimens here. It seems to be like, all the Australian specialists get to show us stuff. I like it.
Yeah.
Okay. I'm trying to leave a lot of the stuff with matter for Lucas, but I will show you this one because they've got the most bizarre inside. All right, that's the inside of a matter. Okay.
Yeah, that's.
And these are the rib heads. And what is inside that, all the way down, is the longissimus coli muscle. And the longissimus coli muscle, we have them too. They make you have the ability to twitch your head sideways. The longissimus coli muscles in chelids, and this is why chelids are the ones that can do long neck. No other group can do long neck because of some things that chelids have done.
Uh-oh. That's the bane of podcasting. You never know who's going to show up. Scott's in a working lab, so.
Yeah, it's in the lab. Sorry, it's noisy. Okay. The longissimus coli muscle is used for striking. So any of the long necks have larger longissimus coli muscles to shoot the head out. And how they do it is they actually pull on both muscles at the same time. And when you pull on both muscles at the same time, it straightens the neck. You got toys like a magic wand, you know, you pull the string and it straightens. Okay, it works on the same principle. That's the engineer in me. You always look at how things work. And anyway, they also have elongation of the skull itself, with the exception of the matter. Because the matter, you know, that's the skull of the matter. It's much shorter. But with the Chelidiners and Hydra Medusa, they have elongation of the skull, which also gives you more length. And of course, that huge gular modification for opening up the throat as they pull the fish in. And the complete combination of that gives them this ability to strike incredibly quickly at prey and engulf it effectively, and swallow it whole. They don't chew, they just swallow. Their mouths actually don't completely meet. So it's a bit like a baleen whale where you've got a mouth full of fish and water. They then close the mouth, close the gullet, all the water comes out, but the fish is held in. And then they swallow. They don't swallow the water as well. It's called strike and gape feeding. It's not unique to turtles. Pelicans eat the same way, so do darters among birds. So it's used in a number of groups of animals. And the thing they all have in common is long necks, flat heads, and elongation of the head, and huge throats.
That's pretty
What
interesting.
you're saying is, like, oh yeah. Were you saying something, Michael?
No, go ahead. Go ahead.
Oh yeah, you're saying is they've perfected that form of feeding essentially, in that I've heard it referred to as the gape and suck method, to where I mean, even in some of the larger soft shells have it. There's a massive, the structures in the throat are much larger, and the jaws are not. There's no crushing power or anything. It's all centered around that gape and suck, and then they vacuum out the rest of the water, or like expel it through the mouth. Right.
You can get bitten by a fully grown Chelidina expansor, and it'll hurt a bit, but it'll hardly break the skin, because they just don't have any closing power in the jaw. It's not meant to. They just want it to be closed enough that the fish can't escape, but the water can. And it's extraordinarily fast. But it is also different within the Chelidina. You've got 3 groups within the Chelidina, for example. You've got the true Chelidinas, like Chelidina longicollis, and then you've got the odd one out in Western Australia, Chelidina oblonga, which is in Macrocephaly, and then the rest of them, like Expansa, which is in Chelidira, the 3rd subgenus. And all 3 of those subgenera feed in a different way. And I had 8 of the species of them, and I tested them all in aquariums with goldfish to see who could hunt the best. And definitely Chelidina barringangii, I had it on any others.
That's interesting.
And
that was in the description of that. The head characters were. Oh, go ahead. The lag here.
Yeah, I was going to say an earlier point. What is it about Chelids that allows them to take advantage of this compared to like the long neck morphology itself? Like, what is the basis for that in general? Like the.
All right. In cryptids, your neck is hidden neck turtles. The neck is pulled back in the vertical plane. Because of that, there is a limit to how long the neck can be. Otherwise, it can't pull it back between the carapace and the plastron. The reason trionychids are the exception is because their plastron is not actually connected to their carapace. So when they strike and then pull the head back, their plastron pulls away from the carapace.
There's like kinesis there.
There's kinesis, but there's no way that, say, even your chicken turtles in the US, which have a relatively long neck for an amidid, they can't do what chelids do. They're not. It's nowhere in the same plane as them. But it's literally just there's no space. between the plastron and the carapace to vertically pull a neck back in that direction.
That's a pretty interesting thing. Just the dynamics and biomechanics behind it. It's certainly an interesting feature. In the description of the Arnhem land longneck, you use the head morphology to describe the species, I seem to recall, and that actually had a lot of predictive power over where they would get assigned in the canonical space, right? But maybe, I guess, just switching to something you mentioned, the Southwestern longneck, that's been nomenclaturally one of the most interesting, one of the most interesting things. I think I've seen maybe 6 different names applied to that, but even subgenre, right? So many different kind of classification. Maybe you could give us the most recent rundown on that. Maybe you could tell us a bit more about this.
All right, and some of that was my fault. What happened with that? John Cann, back in 1977, had mentioned that he was a bit worried that maybe the name was wrong, and I followed that up around 2000, and I examined the holotype and stuff, and I changed it so that Oblonga went up to the north. And the thing I did at the time, which I wish people had followed, when you make a dramatic change like that and you need to go to the ICZN, you can invoke that nomenclature must stay the same until the ICZN makes a decision. Unfortunately, people didn't listen to that. I asked them to do that. I actually invoked it. It's called Article 82 of the code. And so that would have left Oblonga down in the southwest until the ICZN made a decision. Unfortunately, people went on their own track and did whatever they wanted. And so Oblonga started getting used up north, and Rugosa got sunk, and Chelidae got resurrected for southwestern Australia. Then we did the molecular analysis in 2017. I was on that paper as well. And we tested the ancient DNA of 19 holotypes, including the Oblonga holotype, and found it was actually from Perth. And so we had to redo it all again, and that brought Oblonga back in the southwest. That's fine for the species, but we had another issue, and that was the subgenus. Some people refer to it as a genus, Macrochelidina. When Wells and Wellington described Macrochelidina, they set Oblonga as the type species of Macrochelidina. And that the name always goes with the type. Wherever the type is, that's where your name goes, and you can't change that. And so while Oblonga was thought to be what you're now calling Rugosa, Macrochelidina applied to all those northern longnecks. But as soon as Oblonga went back to Perth, Macrochelidina had to go with it. And so that created a major issue in nomenclature. So the correct name for the southwestern one is Chelidina Macrochelidina Oblonga. That is its name. It will not change again. The correct name for the northern longneck is Chelidina Chelidera rugosa, and that will not change again.
That's good to hear that it's stable now. But I mean, that's kind of I guess part of the fun and maybe the frustration of nomenclature and systematics is that kind of, things have to follow a certain order to make it ruly, but at the same rate, it makes it challenging sometimes. You pointed out in one of your papers a difference between taxonomy and nomenclature that I think was kind of an interesting thing that we maybe don't think about the nuances of, but actually has pretty big implications in terms of how it's regulated. Maybe you could talk about that a little bit.
All right. Taxonomy is basically the study of the relationships of species and or populations. It doesn't really matter. But how different populations relate to other populations, and then we draw lines and decide things are species or genera or whatever. You can draw your lines wherever you like. But the taxonomy It's just the study of relationships. You don't have to put any names on anything. You can call them A, B, C, D, if you want. It doesn't matter. And all you're looking at is how different populations are related to each other using science. And it is a science. It's a hard science. And by hard, I don't mean difficult. Hard science means it is a data-driven science, not a subjective one. Nomenclature is a tool. It's not science. Nomenclature is a tool for a bookkeeping tool, effectively, for deciding which name to use for which taxa. And the only place it overlaps is at the type. So at the species level, that's the holotype. The only animal in a population that absolutely has the name of whatever you're calling that species is the holotype. Every other member of that population borrows the name from the holotype. They don't own it. Only the holotype owns the name. So that one singular specimen that is the holotype has the name. You decide that it belongs to that population, so you use the name for the whole population. And that's effectively what you're doing. So when you're doing taxonomy, if you're going to impact nomenclature, you have to study the type. You must include the type in your data set. It has to be there. Otherwise, you can't say anything about the nomenclature.
Right, that makes sense. Ken, are you trying to say something?
I was thinking mostly that another good part of this taxonomy or nomenclature debate is the usage of trinomials, right? Like traditionally trinomials have been resorted to subspecies, but now recently there's been proposals to be using it as like species within a species. Have you heard about that?
Species within a species, well, that is
Yes.
a subspecies, basically.
Yeah, because it's basically a species. It's a subspecies.
Well, the thing is, it depends on what people want to draw the line. In the end, you've got your tree and you're basically drawing a line through it. And you're deciding everything below that line is a species. And so where you decide, it's subjective, okay? Where you decide you're going to draw those lines is where you're going to name things. And so a clade might be a species with a whole bunch of populations that belong to one species, or it might be 5 species and the node that joins the clade is a genus. And that's totally up to the person who did the phylogeny. And usually, if they're doing it well, they will look at how much significant difference there is between them before they decide where to put those names. The thing with species within species idea is basically this is where people are saying there are hidden species within a species and a species should be split up, but it hasn't been done. What people should do is to avoid confusion, if you're not going to work on the nomenclature and taxonomy of a group, you should use the accepted taxonomy. So go to the TTWG checklist, that's your names. If it's not in there, don't use it. You can go to reptile database, go to Wikispecies, where I'm actually an ombud for Wikimedia. And what you are doing is you're not creating confusion by doing that. You're just using the accepted names that are clearly being analyzed as being valid and you're accepting them. If you're writing a taxonomic paper and you want to change the nomenclature, you can do whatever you like, but you've got to provide evidence for why you're changing the names. But if you just start using a name because you would prefer to have population X have this name because maybe you can get more money for the turtles if they are actually a species, that creates problems and you've got no justification for doing it. So unless a species has been analyzed and accepted as a named taxon, it shouldn't have a name.
Yeah, I mean, we're moving towards like the general lineage concepts. At least that's like the trend for these evolutionary studies. But like you said, it's not clear or objective in which species criteria we take to actually define a species, like, I don't know, like monophily or just reproductive isolation. And so there's always this element of subjectiveness. But I think it could be useful to at least propose using trinomials to define species that are not totally reproductively diverged. That's what I've seen.
Most species are not reproductively diverged. Like I said earlier, Chelidae can cross with anything, including other genera. So can many trichemes, so can many sea turtles. They're crossing genera. I'm actually more surprised when turtles can't hybridize.
Right. I mean,
That's a good point.
it's that question of the allopatry versus symmetry too, right? There's certain circumstances where it's easy to see if something can hybridize and produce fertile offspring, but in most cases, that's not going to be something that you can do. Like the Alsaia is a good example. You've got separate river drainages for each one, and they're very similar but different morphologically. So then you have to create a phylogeny. That's kind of your best way to do that.
You've got to take into account with turtles how they move. In many freshwater turtles, the genes follow the water. Wherever the river goes, that's where the genes go.
True.
If there is no way for a turtle to get from river A to river B, then there is no gene flow. And most turtles, freshwater turtles, get out of the water for 3 reasons. They are basking, laying eggs, or dying. That's the only 3 reasons they get out of the water. They're not getting out to walk 500 kilometers to the next river over land. They will not do it. And they can't. And I mean, in a country like Australia, Australia is an extraordinarily dry country. And it has a fair few rivers, but the distance between those rivers is up to 50 to 100 kilometers up the east coast, for example, between each river. And in between the rivers is virtual desert in a lot of parts to a turtle. I mean, it's dry grassland, dry eucalyptus forests, things like that. It's not sand desert like out in the middle of Australia, but it's way too dry for turtles to walk over. And so there's no exchange of genetics between those rivers. The only time the Alsaia exchange is whenever we're in an ice age. Because during an ice age in the southern hemisphere, we don't get covered in ice like you guys in the northern hemisphere do. It gets very dry. And sea levels drop and the rivers connect. Because of the continental shelf, for example, the Mary, Fitzroy, and Burnett rivers, where Alsaia albigula occur, are all connected at the continental shelf. So when water level drops, they're one river. And that's why you get them in all 3 rivers. And so if you've got uplift of mountains or anything like that, all of it comes into bearing on how these turtles distribute. And that's very important in South America, is uplift of the Guyana Shield, uplift of the Brazilian Shield, splitting of the Orinoco into the Amazon. The Amazon didn't exist originally. It's a fairly new river. It only came into existence with the uplift of the Guyana Shield 60 million years ago. Before that, there was just
Yeah, Guyana Shield is very interesting. That whole that whole physiographic region, it's a fascinating place with a lot of endemic species. I seem to recall. The New Guinean turtles are pretty interesting. And that's something that you've dealt with a bit with the Honorachelles. Do you think that there's any sort of cryptic diversity there that's been overlooked, or what do you think that the current state of the taxonomy there is with your Chelids?
It's fairly good. with the chelids, I mean, I think Keratocheles is not one species.
Okay.
But I think
I
it's
remember
true.
you saying that to me a long time ago.
I think the recent paper that described, which was it, that Meaty used when he described that southwestern new species of Alcea, I think he put that in the wrong subgenus because I've got a skeleton of that and it looks more like a true Alcea than a Hanweracheles. But it's a species. I have no problem with that. But I just think he's got it in the wrong subgenus. The Amadura is a problem. And I mean, people are putting up pictures all the time online. I identify turtles all the time online because I can ID about 270 species of turtle by site. And that's what 5 years at CRI teaches you when you ID
Right.
16.
I imagine.
Yeah.
You learn how to ID
Yeah.
turtles. So I tend to ID lots and lots of turtles every week. And I also do it for enforcement when they get custom seizures and stuff like that. I ID all the turtles so that they can do prosecutions. And the Amadura is an issue. People put pictures up of these things called Amadura ganellae and Amadura subglobosa, and I'm not entirely convinced. There's two species there yet, but we'll see.
The Keratocheles, and then we can start to wrap things up and do our little trivia. We like
Yeah, I
to
was
keep
about
it
to bring
there.
that up.
Yeah. Go ahead with
What's-
that.
Because I just, that's one thing that stuck with me that for the last 3 years, I thought of that when I first came to CRI, and I mean, you were showing me around and everything, and I don't even know, through conversation brought that up. You were like, oh yeah, they're most likely, there's cryptic diversity there with the Keratocheles. Is there any significant morphological differences between them, or is it mostly more on the molecular side, all over the board?
It's a little bit of both. Brett Schaefer did do some molecular work on them, but he wasn't 100% sure of the locality data behind it. So he was a bit unconvinced of what to do with it, and so he hasn't done anything with it that I'm aware of. But he found 20% difference between Northern Territory and New Guinea. To put that into context, the difference between Rhodini and Nova Guinea was 7%. So that's nearly 3 times more different than those two. When I looked at morphology in the around 1998, I think I was doing it. I actually wrote a paper describing the entire skeletal morphology of Keratocheles, and I haven't published it. I never published it. And Gene Gaffney looked at it and he thought it was good, and I should have published it. But what I found was that the Northern Territory form, the one in the Daly River specifically, was different to the one in the Alligator River and New Guinea. So Australia actually has both species, I think. But and then you've got one in New Guinea and the Alligator River in Australia. The difference is, first up, the Daly River form does not have salt glands behind the eyes. So there's a pocket in the bony orbit where there is a salt gland in Keratocheles in New Guinea. And this is why the New Guinea Keratocheles can go all the way down to the brackish areas to nest, because they can actually handle being in salt water. The Daly form only sits in the freshwater sections of the rivers. It has no salt glands and no capacity to handle salt water. When you look into the nose of a pig-nosed turtle, if you hold one up straight and just look straight into the nose, the form in Australia, in the Daly River, it looks like a keyhole shape if you just look straight into the nose. Whereas the one from Papua New Guinea looks more like a figure 8 on its side, so it's the infinity shape. Just looking down the nose, and when I look at the bones, there is actually differences in the osteology within the nasal structure of both species. And the Australian form has the white blotches all down the side when they're young. The New Guinea form, that disappears much quicker. The front shape of the carapace is different in both taxa as well. So yeah, I think they're different species. And I mean, Chelidae is actually an enormous family. Chelidae, it has 20 described species in 4 genera. Just Chelidae and Sculptor is the only living one.
Right. That, I guess that's maybe a good last question. The fossil diversity of chelids. What are there whole separate kind of clades of extinct chelids that branch off from the 2 sort of South American Australasian divergence, or are there certain fossil species that are nestled within the crown clade, I guess? How does that look?
It's a bit of both. In South America, there are some stem chelids, which is why I feel they evolved there. There's quite a number of taxa, around 30 taxa, I think, from memory, in South America that are fossil only from the stem. And then, I mean, even matter, you've got Chelus columbianus, Chelus lewisii, both described species, and then you've got Fimbriata and Orinocoensis are living. There's Frenox paranaensis, which is a described fossil Frenox. And I recently found, when I was looking through Musa here in Manaus, fossils of mesoclemens in amongst the collection, along with their other stuff. They've got Frenox and matters as well, but I also found mesoclemens in amongst it. So there's quite a lot. Australia's similar, except most of it's in the crown, but some of it's very old. There's some specimens I mention in some of my papers. The reason I want Myochelys to actually be further split, Flaviemys I think should be actually recognized, is that there is a fossil between Flaviemys purvisi and the rest of Myochelys that is 106 million years old. So the difference between purvisi and a Myochelys is greater than the entire diversity of modern birds and mammals in age.
Wow.
Turtles are old, and this is what people have to get into the head. They've survived 2 mass extinctions, not just the Cretaceous one. They also survived the Triassic one. And we humans are closer to the extinction of dinosaurs in a timeline than dinosaurs are to the rise of turtles. That's how old they are. They're 250 million years old, not million, billion.
That's a really good way to put it. I've never heard someone put it like that. I think that's a good bit to chew on as we kind of wrap up here. We've got a few listener questions, so maybe we could do kind of a rapid fire answer, and then we can go into that trivia. So the first one was, are bright orange Phrynops tuberosus from Guyana real?
I think the Frenops in Guyana are a new species. I don't think the tuberosus. In my view, tuberosus only occurs north of the Orinoco River and only in Venezuela. I think anything on the Guyana Shield is actually a new species, but that's not tested, so I don't know. It
would seem to make sense given that region. We've talked about that a little. And then the second one was the validity of Mesoclemys wormithi, which that recently proposed.
Yeah, that's a complicated one. I'm not a huge fan of them doing it yet. It may have eventually been something that should have been done, but I don't think they had all the evidence to do it yet. But yes, wormithi is the Western form of what people used to call Hanaceps, and now Hanaceps is, Eastern form. But yeah, that's valid. It's what's in the current checklist.
Okay. And then the third one we've got, what to do with all of the old names of types of chelids that are floating. We kind of addressed this, but maybe we can put a spin on this and say, I know that, Scott, you've done a lot of traveling to go and see type specimens. What has that been like? Like that's got to be a cool experience.
Yeah, my first trip in 1997 was a $50,000 trip. That was in 1997.
Dang.
So that's
Yeah.
And I did 2 round the world. I bought 2 round the world tickets to do it, and so I did 2 laps of the entire planet with 10 stopovers off each of those round the world tickets, and that got me to most of the types, and I'm still doing it. I actually am going to see the holotype of Penemes hufibes in about 3 weeks in Munich.
Wow, that's a cool thing to go and see all these different museums and the collections they have. That's hopefully something we'll all get to do at some point when we kind of get through college here. All right, so we've got more questions, but we'll address these later on because we've got some more fun kind of conversations planned for tomorrow and Sunday. But we could just do some trivia now, I guess. We can kick that off. In the interest of time, because we were a little delayed, normally we do kind of a volley here. Well, we can ask you questions, you ask us questions, but if you just want to hit us with a few questions, we can finish this up.
Oh, I sent them to you earlier. Hopefully I can remember them because I'm on my phone, so it's hard to look at my phone.
Right.
It's flipping out. All right, I think I asked you, what was it? One of them was the importance of macrocephaly. Why is it ecologically important in turtles? What is the benefit to the turtle to develop macrocephaly?
Jack is a big enthusiast of this, so maybe he wants to answer this.
Yeah. Well, it enables, they are able to specialize in feeding on extremely tough-shelled organisms. Most of the time, mollusks, but crustaceans and things as well. And the food source that is lucrative in terms of nutrients and ear volume that they can get through that. And they're exploiting a resource that other turtles and just other organisms in the same environment can't exploit. Like the amount of, even proportionally to them, the amount of strength that is required to consistently break through those mollusks is not considered, might not even be, it's not possible for most other creatures to even do.
Humans can't do it without a rock or a knife. You're right. And to give you an idea on that, or just how much power they actually can develop. Emadurum victoriae, when they're fully grown female with massive macrocephaly, can actually grab the freshwater mussel from the Daly River, which grows to around 15 centimeters across that mussel. It's a giant mussel. And they can open those. And what they do is they actually bite the shell only on the hinge where the muscle is that actually holds the shell closed. They crack through that and the shell just opens up. And then they have a 15 centimeter muscle to eat. Quite a feat.
Even these little, this is just a Sternotheris, a loggerhead musk turtle. And you can see the alveolar surfaces they're so broadened, and not even that but this is really difficult to see up close, but the keratin is extremely thick, and it provides but there's also a lot of dexterity in it too. It's not blunt power, like they're extremely precise with applying that strength.
Yes, they are. They apply it to exactly the right place for that muscle to basically fail.
All right, that's a good first one. You got a few more for us?
What was my other one? I asked for.
I see. I've got him up here. I can the first one. I guess I can read your questions. How old is the family Chelidae based on the fossil record? So I'm thinking it's like something like 110 to 100 million years old was the oldest I seem to recall. So Middle Cretaceous, but the split based on the dated phylogenies.
You want me to answer? The oldest fossil. The oldest fossil is 145 million years old.
Okay, all right. Okay, that's interesting because I seem to recall the split for a lot of the phylogenies was about there for the family Chelidae. So I didn't realize that.
This is one of the issues I have with a lot of the molecular phylogenies. They date them using their algorithms, and I see them and they say, oh, Chelidae in Australia are 50 million years old. And I say, but I've got a 106 million year old fossil sitting in my hand. Add that
Right.
up.
Yeah, that
So-
doesn't really work.
It doesn't add up. And a lot of the molecular dates with turtles, I think, are out by nearly double because of the way turtles evolve is about tenfold slower than other organisms.
Right. That's good. Yeah, and some of the methodology, the tip dating that the Holly paper did, I was looking a little into it. It seems like it's sort of a newer methodology. There's a lot of the time intervals, the deviation was like 30 to 40 million years. So that's a pretty big interval. But
It's a
yeah.
time.
Yeah, no, that seems like that maybe we need to narrow that down a bit more. And then they did incorporate fossil material.
He did, because his supervisor was Juliana Sterling, who is a Argentinian paleontologist. So he was always going to be using a lot of fossil material.
That's good. And then doing that total evidence matrix with the tip dating, it seems like it's kind of a challenging thing. How do you actually incorporate that? That
it
makes
is
it more. That's the reason most people avoid the fossil calibration too, right? Is it adds this layer of complexity that a lot of people don't want to deal with. Is that right?
It's not so much that it adds a layer of complexity; it adds a layer of
ambiguity.
Ambiguity, and
Right.
so it reduces your p-value effectively. You know how probable it is that you actually are somewhere in the ballpark of what it is you're trying to say.
Okay, yeah, that makes sense. Okay, maybe one more question. This one was the benefit of embryonic diapause. That was your last one that you sent to me.
Mhm.
I don't know if someone wants to. I could take a stab at this. Ken or Jack, you want to go? Well, so typically it's used to offset the period of time where it's not optimal to hatch. So typically at the onset of the productive season is going to be when hatchling turtles want to enter the water because it's going to be most productive, and that gives them the best fitness advantage for going through the juvenile stage.
Pretty much. Basically, one of the species where I looked at it in, when I was a student, as you know, I was student of Arthur George's, and he did a lot of work on temperature-dependent sex determination. And so I was running his incubators as a job, so to speak. So that was my work when I was, lab tech to be a student. So I had money. And anyway, one of the things we found was that with, say, Chelidina expansa, which has embryonic diapause, they would lay the eggs before winter and get some development done, shut down during winter, and then start again as soon as winter things started to warm up again, so that during spring they would hatch earlier because they'd already got some development. So it gives them a head start effectively in the summer period, so in the productive period, as you say. It's a head starting. Basically, it's a form of head starting for the turtle. Right.
That's an interesting thing that they do. One of many very interesting chelonian feats that there's still a lot to learn. Yes. All right. Well, I think we can finish up this episode, but we've got a few more discussions coming up tomorrow and Sunday. So for listeners, be on the lookout for that. But for now, thank you, Scott, for coming on and talking to us today. And I think I've certainly learned a lot in the biomechanics. I didn't even expect a lot of that to be talking about that, but that's going to be really interesting. A lot of different, the feeding mechanisms and such, and just the classification mechanisms. I think it's been fascinating. So thanks for coming on.
Not a problem. Thank you for having me.
Thank you. It was a great time.
All right. I'm going to end the recording now. Thanks for coming.